Lab 3, Absorbance and Fluorescence. Our main problem was to experimentally determine the dye used in the cuvette. We calculated the unknown dye using Beer's law and an absorption spectrum. Next, we used a fluorescent dye to create an absorption and emission spectrum. Our results for part 1 showed the identity of our dye was the Lepidus dye, and for part 2 it showed that fluorescent had the same absorbance peak but a different fluorescence peak. On the right shows our experimental setup. Our first model is how to identify an unknown substance using absorption spectra. An absorbance is the amount of light absorbed at a path length through the liquid. Our path length was constant throughout the entire experiment because it is the same for every cuvette we used. Scientists use this technique to quantify absorbances of known solutions, and therefore unknown solutions can be compared to these known solutions to determine what the unknown dye is. To calculate concentration, Beer's law was used. The equation for Beer's law is on the right. Beer's law relates the amount of light absorbed by a sample to the concentration of the absorbing species. In summary, as long as we know the distance the light traveled, the concentration can be calculated based on the absorbance. Absorption spectra shows the amount of light absorbed by a solution. We can compare the emitted light from a sample to emitted light from the light source in order to find the difference between them to calculate the absorbance of the sample. A use for this is to experimentally determine the concentration of a sample, and again, using Beer's law. A fluorescence spectra is different than absorption spectra because the wavelength of light that goes into the sample is not the same wavelength of light that exits. Even if the wavelengths are the same numeric value, they are not the same light. A white light source or a UV light is used to excite electrons in the sample, and when these electrons fall back down to the ground state, the energy difference causes a release of light. Different colors of light can be emitted depending on the energy release when electrons fall back down to ground state.
For the experimental setup, we had part 1, which was the unknown dye, and part 2 was the fluorescence. So for part 1, we placed the cuvette between the light source and the optic cable, and it went directly through the cuvette. And for part 2, we measured the absorbance and the fluorescence. So for the absorbance, it went directly through the cuvette, and then for the fluorescence, it went through the cuvette at 90 degrees. Because the flashlight was aimed 90 degrees through the cuvette, for the fluorescence, it wasn't picked up by the optic cable.
For the data and calculations for part 1, we measured the intensity of the water and the purple solution. We did this using the experimental setup discussed previously. Then we used a variation of Beer's law to determine absorbance, which is calculated to be the log of the intensity of the water divided by the intensity of the solution. We then plotted each of these points, comparing wavelength versus the absorbance of the purple solution in the spectrophotometer. We identified our solution as Lepidus dye. This is because the wavelengths match the range of 530 to 570 nanometers provided in the known absorbance spectra. Using Beer's law again, we determined that the concentration was a molarity of about 6.168 times 10 to the negative 6. And here we could see the data and calculations for part 2. To calculate for the absorbance of fluorescent, we simply shined a light through the cuvette containing the dye and measured the transmittance of the light. We then divided the intensity of the light shined through the water cuvette by the intensities measured from the green fluorescent dye. Finally, we took a log of those values to get the absorbance for fluorescent. To get fluorescence, we turned our flashlight 90 degrees relative to the original position, pointing through the cuvette, in order to eliminate the impact of absorbance on the reading. Because the values were so high relative to the absorbance values, we divided fluorescence by a factor of 100 so they could sit on the same graph. On the upper left side, both the absorbance and the fluorescence graphs are visible that we gathered through our data, whereas on the right, the graph provided from our lab manual is shown. What we can immediately see is that the absorbance graph approximately matches the 475 nanometer wavelength peak that is on the expected graph. On the other hand, our fluorescence graph doesn't explicitly show the same peaks as on the expected fluorescence graph. However, if we graph the fluorescence on its own, we can see a slight increase around the range of the wavelength, about 525 nanometers, that we should have expected. We believe that the data is just a bit noisy at this point because the ocean optics cable was too far away to pick up a clear reading. If the optic cable was closer, we suspect that our absorbance and fluorescence graph would be much more similar to the expected graph on the right.
So for possible errors, the fiber optic cable, it was actually taped down, but it was still trying to bend a little bit. The flashlight's battery died halfway through our experiment, so we had to redo part of it. The cuvette, the light did not pass through it exactly the same for every single trial. For the fluorescence, we did not move the ocean optics cable close enough when measuring just the fluorescence, so the reading was a little noisy because of how dim the fluorescence was.